Hello, everyone. I'm Jianzheng Wu from Randall Halfmann's lab at Stowers Institute for Medical Research. In the past decades, various physical states of protein have been observed. Here, I'm showing you an example first, which relates to ALS. On the far left, there is a pool of diffuse proteins tagged with GFP. The diffuse protein monomer can condense into liquid droplet, which can continue to solidify into a gel or transit into solid amyloid aggregates. All these states are different in their material properties and have different effects on their physiology and pathology. Accumulated studies suggest that in addition to the amyloid, studying the entire amyloid process or the kinetics is more critical to understanding their roles in both function and disease. However, studying the full spectrum of protein aggregation is very challenging. If we can detect the assembled proteins such as pre-amyloid or amyloid species for each cell, then taking a snapshot of all cells will give us a better distribution or kinetic information of the entire aggregation. With all these considerations, we developed a FRET-based flow cytometry approach called DAM-FRET to quantify the protein self-assembling at different concentrations in living cells. Here, we tagged our protein of interest with only one photoconvertible protein, MUS 3.1, which can serve as both donor and acceptor. After controlled photoconversion, we can get a mixture of the red form and green form of the MUS 3.1, which can be used for FRET measurements. For the sake of time, I only included the data for the C-terminal domain of the TDP-43, which is a key player for its aggregation. The C-terminal LCD contains a small hydrophobic region that forms ARF helix. Here is a density plot showing the distribution of the cells expressed in TDP-43. Each tiny dot represents a cell. The y-axis is ratiometric FRET, showing the extent of protein assembly, and the x-axis indicates protein concentration. This plot is a snapshot of all cells with different assembled states. We observed here three main populations: the monomer population, the oligomer population, as well as amyloid population. Even though under regular microscope, the oligomer population might look the same as the monomer population, the DAM-FRET we use here clearly separates these two populations based on their FRET value. The relative distribution of these three different states are clearly shown here in the plot. To determine the sequence feature, we have made numerous rational mutations and checked how they affect its aggregation. We found that the ARF helix is a key regulatory element for the aggregation. When we introduce the mutation that disrupts the ARF helix, the amyloid formation is massively increased. In contrast, when we enhance the ARF helix, the amyloid formation is greatly reduced, suggesting that the ARF helix inhibits the formation of the amyloid. We also found that the coalescence of TDP-43 is negatively correlated with the extent of the ARF helix. As you can see, when ARF helix is disrupted, it dramatically induces the formation of the liquid droplet. The second interesting finding I would like to share is that we identified an amyloid inhibitory region with only 3 residues, namely REP loop, as shown here in the red dashed circle. As you can see on the right, whenever we introduce mutations on any of these 3 residues, these all dramatically increase the amyloid formation. A closer look at the structure of the TDP-43 further indicates the potential importance of the cis versus trans isomers of the proline in TDP-43 aggregation. Given that C9orf72 mutation, poly-PR, and proline isomerase, or PPIA, can all induce TDP-43 aggregation. Currently, we are investigating the role of cis versus trans protein in TDP-43, which we believe will provide a better clue to explain the TDP-43 aggregation in relationship to these disease cause factors. Finally, I'd like to thank all the people here and Institute for their support. Thanks, organizer, for this meeting, as well as your attention.